on now. I found that, I'm just going to say yes to this. I found that it's useful to see the paths that people have taken in science. So I'll start with that. And if you can't hear me, and you're free to interrupt me if you have burning questions. I'll kind of pay attention to the chat. And you don't have to be brave to ask me questions in the end in person, I hope. Okay, so I'm really excited by hummingbirds. It's really strange that they exist, and I'll tell you about why. I started not like this. Like many of the people at the Lab of Ornithology were like birding when they were 3 years old, including some of my undergraduate students. And I feel very inadequate sometimes when I see how much they know. But I definitely didn't start my career like that. This was not me. I, in fact, spent most of my childhood not knowing that you had to choose a profession and just rebelling against this idea that you had to do one thing. I was like, I want to be a teacher and a theater actor and a pilot and a psychiatrist and like 20 other things. And my parents were like, you have to choose, like you have to specialize in one thing in school. So in the 11th grade, I chose biology. And then I also wanted to be a psychiatrist really badly. So I applied to like a bunch of med schools, which you do immediately after high school in India. Didn't get into any. So I ended up doing a bachelor's in zoology. And so a friend in my second year of my bachelor's told me that you have to like start building your CV. And I didn't know about that concept before. So it was a transformative idea to me. And since then, I do things that interest me, but also think about my CV in a very, very constant way in some ways. So I started these internship experiences. The first one I had was in this remote site in Assam, which is in the northeast of India. And I studied these Hulaf gibbons for 6 weeks. This one female was being rehabilitated into the wild. She'd been orphaned because her parents had been poached. And so I was studying her behavior before and after her release into the wild, where she was paired with a wild male. And I just took a bunch of photos of things all the time, especially because I only had a small like point and shoot macro lens. I only looked down. There was no point looking up at things that were far away, like birds. And so I never photographed birds. I only photographed butterflies and snakes and reptiles and things that were really close to me. And those are the taxa that really I enjoy the most, honestly, still. I then did an internship where I tracked king cobras for a couple of weeks in South India with this radio telemetry program, which was fascinating. And then I went on to do a master's and study hornbills, which are these giant toucan-like birds in the old world in Africa and in Asia. And I was looking at the nest site behavior of this species, the Malabar pied hornbill, in a very fragmented landscape in India. So if you look at my CV now, it looks pretty linear, but at every single stage, sometimes every 6 to 12 months, I go through an extreme existential crisis. Sutirtha witnessed one of these at a conference this summer. I was like, what is the point of anything? Why do I study hummingbirds? Like, who cares what their biology is? Like, the world is burning. But then I come back to being fascinated by it and convincing myself that my curiosity is important and people care about the curious things, while also trying to build a more applied component into my future work. Anyway, after that sidetrack, I then did a few more internships where I studied butterflies and their visitation rates to invasive species. This is one of my favorite butterflies. It's this, let me get my laser pointer. It's this map butterfly in India. It has very translucent wings. It's really delicate looking. It's beautiful. The way it flies is just incredible to watch. So then I applied for a bunch of PhD programs, and that was another lesson in failure. I applied to like 3 or 4 different programs, didn't get in the first time because I had no idea how to apply to things in the US. And then the second time I wrote to like 80 professors, 30 of them replied. I applied to about 8 programs and I got into 3. And 2 of them were studying herps, like to study coral snakes in Indonesia and toad in Lubbock, Texas. But I decided to take the program that I took at Stony Brook because the questions were really interesting to me, even though it was with birds. And so I went to Stony Brook in New York to do my PhD studying hummingbirds, mainly in Ecuador, but also in Arizona. And so that's what I did for the next 6.5 years. I spent about a year total, a little bit more than a year in the field, studying hummingbirds. So this was one of my field sites in Arizona, where they feed on these tiny little red flowers in the foreground in this desert. This is another one of my field sites in Ecuador, the high elevation Andes. And I also had some field sites in the mid elevation Andes in Ecuador. So I loved being in the field. I think it was a very difficult time managing my field team on my own and leading my own field team, but I learned so much about mentoring and managing data and people that it was a valuable experience. I also got to spend a lot of time with some incredible people. This is Doña Rosa, who managed our field site in Ecuador. She just used like a basic phone, like she didn't have a smartphone. It was very difficult to reach her. She had very annoying dogs, but I spent 7 months at that field site, and it was an incredible time. Also, I spent time with Dawn Powers, who's faculty at a small Christian institution in Oregon called George Fox. And he's been my mentor now for 10 years, and I've continued to collaborate with him. And he was key to making me the scientist I am today. So I think of myself as a physiological ecologist. At every career stage, I've tried to learn a new set of skills and new set of methods, because my goal is to do research in India and in the tropics in general. And I think there's a dearth of advanced methods in ecology in many places in the tropics. People don't have the access to the equipment, but also the training to be able to know what they can, the possibilities of what they can do. And so I've tried to learn new skills at each stage, and I'm trying to become a more broad integrative biologist that way. So I can mentor my students and other trainees to use all these methods. And basically the question that I'm interested in understanding is the mechanisms that animals use to cope with variable environments. So hummingbirds have been my focus for the past 10 years because they have really high metabolic rates. They're really small and they barely store any fat. And so they're constantly on this knife's edge between life and death, because if they don't feed for a few hours during the day and they're still using up energy, they can die. So I made this infographic to tell you just how high their metabolic rates are. If an average, like Matt Damon-sized human could just eat potato chips all day and meet all of their caloric requirements, they would need to eat about 15 packets of potato chips a day to survive. And we'll ignore that we need vitamins and stuff, who cares? So then if that same human had the metabolic rate of an elephant and was just eating potato chips all day, they would need about 4 packets a day to meet their energetic requirements. So as you get bigger and bigger in body mass, you need less energy per unit of your body's mass to stay alive and meet your daily energetic needs. Can anyone guess? You can put this in the chat. How many potato chips a human with a hummingbird? Oh, my battery's low. God, that's so embarrassing. Let me plug it in. 1 second, sorry. I was so sure that wouldn't happen. Okay, crisis averted. So that should have given you time. No one's put anything in the chat. How many potato chips? Oh, okay, here we go. 165, 100, 150, 200, 250, 45, 45. That's very low. 200, 1,000, 145. Okay, I think that's enough guesses to form some sort of normal distribution. The average guess is around 150-ish, right? 675 is very, very close, Katie. They would need, a human with a metabolic rate of a hummingbird would need about 600 packets of potato chips a day to meet their daily caloric requirement. And so as you get really, really small, you use way more energy per unit of your body's mass than a big animal. And so this kind of speaks to this idea of allometry, or how some trait scales with another trait. And I'll talk a little bit about that. But given all these things, that this hummingbird is small, its metabolism is really high, it's barely storing any backup energy, it's a miracle that they've managed to diversify into over 330 different species across a range of environments, from the Amazon to the desert to the high elevation mountains. And now there's even hummingbirds overwintering in Oregon and Washington. So it's crazy that they managed to live in all of these different environments given these extreme energetic demands. And so some aspects that I'll talk to you about are their allometry, because they have an unusually high allometric slope. We look at whether their daily energy expenditure is fixed or variable, how much energy they spend in a day. And what do they spend that energy on? The literature shows that they're mostly spending their time sitting, perching, like 70% of their time, but we'll inspect whether that's true across a range of conditions. And then what do they do at night, which is my favorite. I don't have favorites, but this is my favorite. Maximizing topper use is maybe what they do or not, we'll see. So this work kind of covers what individuals do, what individuals across species do, and what happens across species. So we'll start with allometry. It's the study of how one trait like metabolic rate scales with another trait like body mass. And I thought this was an extremely dry and boring subject, and I couldn't possibly have any interest in allometry until it started becoming relevant to my work. And then I read like obsessively about it for weeks. And it's fascinating because the study started in about the early 1800s, when people wondered if there were rules governing how animals scale with size. And one of the interesting applications of it was for veterinary science, because if they knew how much to dose a horse a certain medicine, they wanted to know like how much to dose a dog, for example. And it's not completely obvious if you don't know anything about the principles of how animals scale, right? How do you know what to give a dog? So they came up with these relationships and found and proposed that there was a universal scaling law, that the slope for all of this whole relationship was 0.75, no matter what taxon you were or what animal you were. But later on in the late 1900s and late 1990s and early 2000s, a lot of studies started coming out showing that the scaling relationship really depended on the taxon. And so birds have a different scaling relationship than mammals, than reptiles. The slopes still look kind of similar here, but they can be different. So what do these slopes mean? If you have energy expenditure on the y-axis and mass on the x-axis, these are always log-log scales. A slope of 1 means that a big animal has the same energy expenditure per unit mass as a small animal. If you have a lower slope, like lizards do, a bigger lizard has a slightly less energy need per unit mass than a smaller lizard. Mammals have an even lower slope and birds have a much lower slope. So a big bird needs way less energy per unit mass on average than a small bird. And hummingbirds are birds. So you'd think that they would maybe follow this kind of slope equation relationship, but they do not, at least from this 1999 review, which included 6 hummingbirds among many other species. They found that the scaling relationship for hummingbirds with energy expenditure and body mass was about 1.21. And that's higher than 1, which means that a big hummingbird is spending way more energy per unit mass than a small bird. And this doesn't make any sense for scaling. Like if you're trying to produce a bunch of bags for a factory, if you produce 20 bags and you're thinking of scaling them up to producing 100,000 bags, you want each bag to cost you less to make as you scale the factory bigger, right? It doesn't make any sense to scale something up if it's going to get more and more expensive to be a big version of yourself. So why would a big hummingbird even exist if it was so much more expensive to be a big hummingbird than a small hummingbird? So we inspected this with more species. We added sampling across a range of hummingbird species to test whether this was actually true. Are smaller hummingbirds really and more energetically efficient than bigger hummingbirds? How do you do this though? How do you measure a hummingbird's metabolic rate? You can't use a Fitbit. I use one on myself, and it's a great way to collect data for big animals. But for hummingbirds, the accelerometer plus battery plus storage is way too big, and it would weigh the hummingbird down or fall off. So we catch hummingbirds. We use these feeder traps very conveniently. In some places, at least, they come to feed us. At one of my field sites in Ecuador, we didn't get a single hummingbird recapture in the 7 months that I was there continuously. So some sites, feeders do not work. But at this site in Arizona, we were able to catch birds like this. This is the blue-throated mountain gem on the left and the black-chinned hummingbird on the right. One second. Yeah, so when we catch the bird, one way we can measure its total daily energy expenditure, I think, is this ingenious method called the doubly labeled water method, where you feed or inject the bird with a double isotope of water, and then you let that water equilibrate with its body's water. And then you collect a pee sample, which is like one of my favorite activities, honestly, with hummingbirds ever. I get really excited when they pee because it's like, yes, data, very, very obvious data. So we collect a pee sample and then let the hummingbird go. And in the 24 hours that they're out in the wild before we try to recatch them, they're breathing out carbon dioxide and they're also peeing out water. So they're losing oxygen 2 ways, and they're losing that hydrogen only 1 way in the pee. And the difference between the rates of loss of these 2 isotopes, we collect another pee sample the second day and measure that, is carbon dioxide production. So the difference between the hydrogen and the oxygen gives you how much CO2 they've been breathing out. And carbon dioxide is a really good proxy for the amount of energy they've spent in 24 hours since they've been in the wild, because most of your metabolic processes end in carbon dioxide. And so we can see how much energy they've spent in whatever field site we're at over 24 hours. And so we plotted this with body mass. We basically had a total of 17 species instead of the previous 6. And we looked at, this is species means in the bigger points and the individual values in the smaller points. So it's quite scattered with individual, but overall the relationship was the slope was closer to 0.96, which is less than 1. It's still really, really high, but it doesn't seem as crazy as the previous 1.21. The confidence intervals do still overlap 1, so maybe more data would be useful to figure this out some more. But this is good to know. And so hummingbirds seem to have a scaling slope that's closer to lizards than to that of other birds, which is really interesting for maybe reasons that'll become clearer later, though I won't address them explicitly. Lizards are ectotherms, hummingbirds are heterotherms, birds generally are endotherms. I wonder if that has something to do with it. So scaling depends on what energy is spent on. This is something I learned in that weeks-long literature search that I was doing, and I came across Doug Glazier's papers. He wrote a bunch of papers coming up with this metabolic level boundaries hypothesis, which says that scaling depends on what you're spending energy on. So if within a taxon, if they're spending energy more on muscle-related activity or on torpor, which we'll talk about later, then the slope is closer to 1. And if they're spending energy more on surface-related processes that have to do with your surface area to volume ratio, then the slope is closer to 0.67, like on heat flux or water flux. So this could be related to what hummingbirds are doing. So we looked at then what constituted their daily energy expenditure, what were they spending energy on. The first step, though, is defining how variable that energy expenditure is. And so in one study, we went to 2 different sites in Arizona, which were very close to each other, but quite different. So this is Harshaw Creek. It's very elevationally diverse, vegetationally diverse, lots of microclimates like this, where Becca is taking a nap in the shade, and hummingbirds can do that too. And this is a close by site called Sonoita Creek, which is much hotter, drier, flatter, less vegetationally diverse. Definitely not that much microclimates for us to take shade and rest in. So we looked at daily energy expenditure at these 2 sites, and you can see Harshaw's on the left and Sonoita's on the right. And we studied them. We collected samples from both the pre-monsoon and the early monsoon to look at how this changed across seasons and across sites, daily energy expenditure, which was on the y-axis. And this is with the broad-billed hummingbirds in Arizona. So this is what we found. The black dots are individual measurements of daily energy expenditure, and the colored dots are the same individuals that we captured in 2 different seasons. And so even within an individual, you can see how and whether a daily energy expenditure changes. So some individuals didn't change their daily energy expenditure. Others increased it hugely as the season shifted. But what really struck me about this graph is in Sonoita Creek, in the early monsoon versus the pre-monsoon versus the early monsoon, the points don't even overlap. So the amount of energy that the birds are spending is completely different and much higher in the early monsoon compared to the pre-monsoon. Why? I thought it would be temperature because these sites are so different thermally that I thought it might be thermoregulation that's changing their daily energy expenditure. But we had to quantify why. And so we were looking at what components of their activity budgets and their energy budgets vary the most. How do we do that? So we use this other genius method called respirometry, where you use this basically suitcase, which has sensors in it, and you pull a breath sample from the hummingbird. So you pull a sample of the hummingbird's breath and you measure the amount of carbon dioxide they're breathing out and the amount of oxygen they're inhaling. And you can use that again as a proxy for how much energy they're spending doing that activity. So we can do this while the hummingbird is hovering, while it's resting, while it's at different temperatures to look at thermoregulation, while it's sleeping at night. We can quantify different aspects of its day by doing this. But by doing this, we can measure the energy per unit time of different behaviors, but not the time they're spending on different behaviors. And so that's something we had to model because we don't have the accelerometry kind of data for them. But we know what the total daily energy expenditure is. We know the energy per unit time of different activities. And so we could model the amount of time they were spending on different activities. That was the only unknown part of this equation. And so I'm going to show you a very similar graph to the previous one that you saw with daily energy expenditure and site on the Y and site and season on the X-axis. But this time I'm going to add on our model data. And so the traditional literature values were this model where we had 15% of the time spent hovering, 15% flying, 70% perching. So people with binoculars watching hummingbirds found that they were spending about 70% of their day perching. And this was with more territorial species, just something to note. We also modeled a much lower activity scenario and a really much higher activity scenario, 80% of their day hovering and flying and only 20% perching. So this is what we saw. The gray box plots are from doubly labeled water values, which you already saw earlier, and the colored points are the modeled values. And so there were different activity scenarios that could explain what was happening in multiple of the sites and season conditions. So the birds could have been using this traditional model here in Harshaw Creek, but not really at any of the other sites in the OR seasons. But what struck me was in Sonoyta Creek in the early monsoon, they were spending most of their day hovering and flying. There was nothing else that could really explain this kind of daily energy expenditure. And that's a lot of activity. So thermoregulation and all of the other components are inside this range of variation within these colored lines. And so thermoregulation was really very little compared to completely shifting how much time you were using on different things. And the high activity scenario was the only thing that could explain Harshaw Creek in the early monsoon. So around this time, I started collecting data on myself and how I spent my day because I was very insecure about not working enough. I thought I was being a really lazy PhD student. And so I found many things I did this for like literally almost 4 years. I collected data on how I use my time. Every 15 minutes, I would enter it on my phone. And I found that I spent an average of 5% of my day or less on physical activity. Most of it is salsa dancing, like Jack said in his introduction. And so I spend less than an hour a day on exercise, and hummingbirds spend at least 3 hours a day on exercise, but up to 13 hours a day. 13 hours a day. Imagine spending 13 hours a day every day hovering and flying, which hovering is 10 times as expensive as resting energetically. So they're spending a hugely variable amount of time on daytime activity, but it's also really expensive. And so, yeah, I learned a lot about myself in this process and completely shifted how I use my days. But also I learned that I'm very lazy compared to a hummingbird, but that's okay because their physiology is different from mine. Around the time, this is a complete side note, I started collecting data on my mood. I realized that I could be my own study subject and use data to transform how I understood myself. And so every day, twice a day, I would enter how I felt, like whether I felt amazing or good or meh or extremes or bad or awful. And the real reason I was doing this was to see whether I should marry my boyfriend at the time. because I didn't know how, like I knew I liked him, but I was worried about like the 10-year long projections of how things would go. And I was worried about what his mom would think and blah, 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 like all of these long-term things. And so I started collecting data every day to see like, how do I feel about him in the daily, you know, like, do I feel good being with him? Does he make me happy? And you can imagine I'm sharing this because it worked out. We got married like 2 years ago. I did end up realizing that I'm very happy on average with him. So that's these green bars. This is the number of days at that time, but it ended up being over like 1,500 days that I collected this data. And then this was the next year. I started this postdoc and I was miserable. I was in Alaska for 1.5 years. And that was a whole set of other realizations, which was very interesting. I think it's really useful to collect data on yourself. And most people, this was the time I collected the data for until 2021. But yeah, you can see like patterns on very different scales by doing this. Just a suggestion if you're confused about things and you think an impersonal view of yourself might help you. Okay, so going back to hummingbirds, they use their time very variably, we learned. They can change how they use their day. And what is happening around them that's causing them to shift how they use their time. So we looked to environmental conditions other than temperature, and we found that flowers could probably explain what was happening. The resources in Harshaw Creek and in both sites in the pre-monsoon were much more abundant and dense. So there were more flowers and more clustered flowers in the pre-monsoon and in Harshaw than in Sonoita and in the early monsoon. And so as soon as the rain started, the flower abundances kind of dropped and the flowers were more scattered in the landscape. And so we think that the birds had to spend more energy and time getting between flowers and finding flowers in the early monsoon than before. And so that's been like the whole animal energetic need stuff, but this really got me thinking about what was happening within an organism. I used to wonder what's happening at the organ level to completely shift this daily energy expenditure. And I got really interested in the underlying genetic mechanisms that underlie that energetic flexibility. So the real component of this that I'm focusing on now is the hummingbird's ability to be heterotherms. They can shift from being endotherms, generating their own body heat, maintaining a high body temperature during the day, to being ectothermic at night. So they become like lizards and snakes. They allow their body to get cold, save a bunch of energy, and make it through the night by being ectothermic very often. So they are heterotherms. And it fascinates me that heterothermic animals exist at all. It's such a huge amount of variation, and there's so much changing in your whole body every night sometimes, or for hibernating animals for multiple weeks. That's just such an extreme strategy to me. And so I'm really interested in torpor. And torpor can be daily, which is what hummingbirds do, or multi-day, multi-week torpor, which is the same as hibernation. So there's many ways we can study this. One of them is respirometry, which I showed you earlier by measuring the oxygen and carbon dioxide in their breath. Another way are these thermal videos. So this is a pretty high resolution thermal camera that my collaborator Don has. And we can watch these hummingbirds at night and look at their surface temperatures and how they change. So this is a black-chinned hummingbird in Arizona. This is its eye. This is the beak. This is the tail. You can see the eye looks like it's the hottest part because there's very low feather density around the eye. So we use that the eye skin temperature as a proxy for what's going on inside the body. You can see this bird is kind of moving a little bit. It's breathing pretty quickly. Its heart is probably beating pretty fast. It's maintaining a high body temperature. It's asleep. Its eyes are closed. Later at night, the same bird is in torpor. And this is also a video, but you can see the bird is barely moving. You have to kind of squint to even find the bird. It's in the same place as the other video. But this black-chinned hummingbird is in torpor. Its whole body is at about 15, 16 degrees Celsius. And it's saving like, I don't know, 60, 70, at least 70% of its energy by being in this state. And then this bird warmed itself up the next morning about an hour before sunrise and went about its day, no problems, nothing, not dead, first of all, but also like functioning at its normal levels. So just to highlight what a stark difference this is in what they're capable of, humans maintain a high body temperature of about 37 degrees Celsius. hummingbirds 41 degrees Celsius. You can look at the Fahrenheit if you want. I just think in Celsius, it's really hard for me to convert. So there's Fahrenheit on the screen for the non-Celsius people. Humans, we are considered hypothermic if we drop by even 2 degrees Celsius and go down to 35 degrees Celsius. We have to be externally rewarmed with a source of heat. Whereas hummingbirds can drop, the coldest a hummingbird has been known to go is to 3 degrees Celsius. So they can drop by up to 38 degrees Celsius or 68 degrees Fahrenheit. And they're functioning at about 2 to 30% of their normal metabolism. And then they can warm up and their heart keeps functioning the next day. Their brain keeps functioning. They can remember where flowers were, like no major long-term side effects as far as we can tell. So I'm really interested in studying how this happens. And so now in my postdoc at the Lab of Ornithology, I'm looking at what genes are expressed, because now imagine you're functioning at 5% of your normal metabolic rate. What are you spending that 5% on? That's what I really want to know. Like, what is so important that you cut 95% of your metabolism out, but this has to stay to keep you alive? That's what I'm trying to look at with gene expression. So that's what I'm using the transcriptomics for. We went out last summer and this summer and studied Anna's hummingbirds in Oregon. And so this is Emily Blackwell. She's a student with Dawn at George Fox. So we built these really neat chambers out of acrylic to be able to measure respirometry and thermal imaging data at the same time. And so we replaced this front clear plastic with a thermal window that we can't see through, but the thermal camera can see through, and to get images like this. And so again, this is a bird that's asleep and another bird that's in deep torpor. You can't really see it except for its beak. That's pretty cool here. So what we did was we collected Anna's hummingbirds in 3 different metabolic states when they were asleep, in transition to torpor, and in deep torpor. This past summer, we collected sleep, deep torpor, and coming out of torpor, because that seems to be important in the mammal world. We collected 6 individuals last summer per metabolic state and 4 more this summer. And we collected a bunch of different tissues. We euthanized them in these metabolic states. Unfortunately, I wish there was a less invasive way of measuring gene expression across tissues, but we collected as many tissues as we could get, basically the heart, muscle, liver, lungs, 3 sections of the gut and the brain, and we did RNA sequencing on them. So something we noticed when we were dissecting these birds was that the birds presumably would lower the function of a lot of their organs, but their heart would keep beating when they were in torpor for at least 10 minutes after they were dead, sometimes more. Whereas when they were not in deep torpor, their heart would stop beating pretty quickly, like when they were at a high body temperature. So this tells me very visually that these birds are probably, their hearts are probably really hypoxia tolerant. They're probably not getting that much oxygen in general when they're in torpor because their breathing rates are very low, but they are still functioning at some level. And then if we hadn't killed them, they would have warmed back up and again functioned at normal levels. So here are some of our preliminary RNA-seq results. I found it really interesting that there was this one gene that was significantly downregulated and its expression levels were lowered across all tissue types except for the lungs. I really don't understand what it does. It has like a gazillion functional associations and it's across multiple categories, chemical, disease, functional terms. Transcriptomics is really new to me. So if anyone has an idea of how to find out what it really does with more specificity, I'd love to hear it. But across all tissue types, its expression really decreased. So this is gene counts on the y-axis and the different metabolic states on the x-axis, normothermy, which is normal high body temperature sleep, transition, and deep torpor across each tissue type. So the guts are on the top row, then there's heart, liver, lungs, and pectoral muscle. You can see lungs are the only ones which didn't have a lower expression of this. It just struck me because it seems like a good proof of concept that our method worked and we didn't mix any samples up and it seems to have done what we expected. But we also did some pathway analyses. And so this is using this site called Enricher, where we can compare, we can just take the set of upregulated genes and put it on the site, and it'll tell us what pathways those genes tend to be associated with. And what was really, really exciting for me was that the circadian pathway seems to have been upregulated across tissues in deep torpor compared to normothermy. So they're functioning at 5 to 10%. There's more genes overall significantly downregulated than upregulated. About 70 are upregulated and 100 and something are downregulated. But of those 70 that are upregulated, the circadian pathway seems really important. And so keeping track of time when you're functioning at very minimal metabolism seems key. And this makes sense because when you watch these hummingbirds in torpor, they come out of torpor and start to rewarm almost always about an hour before sunrise, no matter when they went in. Their entry time is variable, depends on their fat stores, but their coming out of torpor seems to be very consistent. And so they need to keep track of time. And that shows here. So some of the circadian genes that we looked at were the clock gene, which is a fascinating pathway that if you know how the clock works, if you don't know how the clock works, there's a central clock in the suprachiasmatic nucleus in your hypothalamus. And it's basically this chemical reaction that happens with regularity every 24 hours. And so that central clock in your brain syncs up with all of the peripheral clocks because you have a mini version of this going on in multiple organs. And those peripheral clocks are told by the central clock what time is. And this is often regulated by light. So yeah, the whole circadian system is really interesting, but in the heart and in the lungs, you can see that this clock gene was upregulated in deep torpor compared to the other states. And the same was true for Cry1 and Cry2, which are also circadian genes, and opposite for PER2 and PER3, which are also circadian genes. So I think they're opposing pathways, but I need to look more at this. And so this is across tissue types. You can see that clock gene expression increased in deep torpor, except in the distal gut, like the hind part of the gut, where it decreased for some reason. And then this is Cry1 and Cry2 across all tissue types. Cry1 and Cry2 increased in deep torpor compared to normothermy. So we're just starting to understand how to analyze all these data, but I have like 3 freshmen working with me, so they don't know how to do this either, and I'm trying to learn and then teach them. But we're looking at metabolism genes, cardiac genes, mitochondrial genes, and fat storage and fat-related metabolism genes, because I think those will be really interesting and relevant to torpor. I'm just going to skip that. I don't know why that's in there. I keep forgetting to take it out. So there's also some pathways that are downregulated in torpor, but it's not as obvious to me what they do. There's keratin sulfate and keratin metabolism. For some reason, that's like the most downregulated thing in deep torpor. And there's post-translational protein modification and some tumor suppressor genes that are downregulated in deep torpor compared to normothermy. So there's a lot of data here. There's like 15,000 genes in each tissue that we have to make sense of. And someone told me it's an art to make sense of gene expression data, which is very confusing because that means that you can't teach me how to do it. I just have to figure it out. But I'm going to do that. So the longer term implications of this are that heterothermy is a strategy to deal with variability, but of the over 10,000 bird species that exist, we know about torpor from less than 1% of them. And so there's some natural experiments that lend themselves to really trying to understand the evolution of heterothermy some more. I would love to go and study sunbirds in their range across Asia, but especially in India, because sunbirds and hummingbirds are both nectarvorous species. They feed on nectar and maybe probably also insects. So they're very convergent ecologically. They're very small also, the sunbirds. But they're passerines and they're separated from hummingbirds by millions of years of independent evolution. And so evolutionarily, they're quite distinct, whereas ecologically, they're very similar. And on the flip side, there's these nightjars, which obviously look very different than hummingbirds. They are big, they're nocturnal, insectivores largely, and they are very closely related to hummingbirds, and they also use torpor. So it would be really nice to compare the potential for torpor across these different bird groups that are either ecologically or evolutionarily similar to each other. And I think these kinds of approaches to studying heterothermy can help us understand the evolution of heterothermy. If we look across birds and mammals, especially at all of the endotherms, and look at whether heterothermy is a shared ancestral trait or is an independently evolved trait. We can also understand more about the evolution of endothermy by comparing birds and mammals. And then there's a lot of interesting applications from this work. So with mammals that use torpor and hibernation, there are these, at least with the hibernating mammals, there are these tau proteins that build up in hibernating mammals' brains. And Alzheimer's patients happen to build up these tau proteins in their brains as well. Hibernating animals can get rid of them when they come out of hibernation, but the Alzheimer's patients don't. We don't know whether this is causative, if the tau proteins actually do something bad, or if they just happen to be a side product of having Alzheimer's. But I think it would be useful to know how hibernating animals are able to clear them out. We can learn a lot about safe surgeries and about cardiac function, I think, by studying animals that hibernate, because they're getting their hearts down to very cold temperatures and also slowing their heart rates down a lot. And then when they warm up, they're not bradycardic or tachycardic, that they don't have irregular rhythms of heart function. So it'd be useful to know how they're able to do this. Immune function is also suppressed in animals that are hibernating. So bats seem to be really susceptible to white nose syndrome, partly because they're very susceptible when they're in hibernation. Their immune function is very suppressed. And you could see how excited I was by the clock genes. They're able to keep track of time. Even these hibernating mammals that are in burrows way underground for 6 to 8 months somehow know when it's time to come up and out of hibernation and mate. When they don't have any light, they don't have access to food, the temperature is very stabilized relative to the outside. So there's a lot we can learn, I think, that's broader from studying torpor. And there's therapeutic implications potentially. There's medical researchers who are trying to cool humans down safely and rewarm them. Because when you cool anything down, any living thing down, its enzyme function decreases, all of its metabolic processes slow down. And so you're slowing down potential damage to the system. So for a stroke or a cardiac patient, you can buy more time to do the surgery by safely cooling them down. But cooling has a lot of effects, like you're going to freeze cells to death at some point and slow down function in bad ways. So studying torpor could help us with that. And then one of my favorite things to think about is the science fiction realm, which currently thinks about humans hibernating. And I was on this very interesting panel where we spoke to science, like people who study torpor and hibernation were on a panel with authors who write about human hibernation and space flight. And basically in all of these books, you're like, human was on Earth, and light years later, human is in another galaxy, and everything is great, voila, everything is perfect. I am the same person still. But that's probably not what would happen. You probably would die. And if you didn't die, you'd wake up with brain damage probably, and I don't think you remember who you are. So it was a really interesting panel to look at the biology of what this would involve, especially with all the recent funding into recent, private funding in spaceflight. It would be really interesting to think about how this could be applied. And I'll slow this down by saying that I love outreach. I can't do science without feeling like it has some broader impact, and that I'm telling people about it, at least, if not having more, conservation application of my work immediately, which is what I hope I will have in the future. But I give a lot of talks. I think this is my 98th talk, and I mentor a lot of students. It really gives me incredible joy and purpose to get other people excited about science, even if they don't want to be scientists. And I absolutely love writing grants. I don't know how common that is, but I hate writing papers. I think writing grants and writing papers are like orthogonal interest sets or skill sets. I think people who like one don't often like the other, and vice versa. I do not like writing papers, but give me a grant and I'll write it any day. I've applied for like 50 or 60 or something, and I've gotten half of them. So I really enjoy it. A lot of positive reinforcement. So I'd love to thank all of the people that have made this work happen, including these 5 students who I'm currently working with. my generous, helpful, encouraging collaborators and mentors, without whom I wouldn't be in science, and the logistics and kindness and warmth of the people at my field sites who have hosted me for months at a time. Thank you. I love your questions.
Great. Thank you so much again, Anusha. That was a really fantastic talk. It's really cool to see all the creative methods and technologies that you're.
Is Jack frozen for other people? Oh.
What?
Hi, so I'd be curious to ask what your perspective is on something. Are you frozen there too as well? Can you hear me?
I can hear you.
Okay. So hi, I'm Ken, and I work on amphibians, salamanders mostly in particular. We've done a lot of ecophysiology of salamanders, like living at different elevations. So thank you for a really fascinating talk. It got me thinking about some things. You know, a lot of this work is still very recent, like in our field, like coming at this from an evolutionary perspective, like what kinds of physiologies should evolve under particular environmental conditions and how long would they be maintained for, whatnot. So there's some literature that was published in the last decade talking about what you're talking about. It sounds like something similar. Of course, we're working with ectotherm, something called metabolic depression, where certain things, so in particular, mountaintop endemics, and even within mountaintop endemics, salamanders that live on the edge of the range show a greater propensity for metabolic depression. And that's been interpreted by some as, as the fact that, well, these mountaintop endemics are melting down on the edge of their range. I wonder I've always wondered if it might actually be, are they melting down, or is it metabolic depression is actually an adaptation to living in harsh conditions and maintaining a range limit, at the edge on harsh conditions. So I wonder if you, thought about any of that in your work in general, and how this stuff might relate to, geographic range and the habitats that these creatures live in.
Does that mean that their like baseline metabolism and their standard metabolic rate is lower on the edges? Is that what that?
Yeah. So they depress their metabolism, so they relative to basically the basal metabolic rate. Mm-hmm. They will be salamanders of the same size on the edge of the range will be basically using less energy per gram than those in the interior of the range. And I guess I'm just curious what you think about it. Like, this has been interpreted to some as like a stress response.
Yeah.
Is it a stress response, or is it an actual, is it like the animal melting down, or is it they're actually adapting at the edge of the range?
Yeah. I think it would depend. I think you'd have to probably measure other things to figure that out and parse it. But I mean, we change our metabolism with seasons. Our basal metabolic rate changes in the summers and in the winters in the temperate regions. Yeah. I think there are lots of, you could argue that we're more stressed when there's less light in the winter. I don't know. But yeah, I think it can definitely be an adaptation. There's a lot of change in basal metabolic rate depending on latitude, depending on, I don't know, all kinds of things. It doesn't necessarily have to be a stress response, I think.
Yeah, I was curious what you thought, if you had thought about it. But
yeah.
Thanks for entertaining the question. Great
Good,
talk.
thank you. Yeah. There's a couple of questions in the chat. Jack, should I take those?
Yeah, go for it.
Yeah, go for it.
Okay. The first one was, Hunter, did you experience any problems with territorial hummingbirds at feeders? Did this complicate the method for capturing a variety of individuals? I did not think of it as a problem, but there were definitely lots of territorial hummingbirds at feeders. There are some non-territorial ones that just like sneak in and sneak out and no one pays attention to them. That happens too. But I didn't really start out by knowing which species I was going to target. I was just like capturing what I could get because I wanted a range of species and a range of body masses. So only more recently have I become targeted with the gene expression work on like, I want this species, I want this individual to go into this metabolic state. I wanted it to happen at this time. Like I have had to be much more predictive about what's going to happen with the hummingbird that I catch. But the territorial ones didn't really bother me. Yeah, in Arizona, we were able to catch a range of territorial and non-territorial ones fine. I think you can, if you put out a bunch of different feeders, and the territorial ones might come to one and not to the other all the time. So I think we're able to catch a variety. Alex said, I was curious about the evolutionary link between nightjars and hummingbirds. I was wondering how nightjars use topper. Also, if from an evolutionary perspective, I was wondering what the MCA of these two groups looked like. I'm imagining something more like a nightjar that started utilizing flowering plants while pursuing pollinators for food. What's MCA? Is it an evolutionary term? I'm ignorant.
I think I'm saying most recent common ancestor.
Oh, okay. Sorry, I feel very dumb. It's very close. So I think the nightjars, the swifts, and the hummingbirds are all from one recent common ancestor. I don't know how old that is. Probably, no, I don't know if I want to say 20 million years, because I know that yeah, I should know that. I don't know that.
So like the nightjars, they're apodiformes, aren't they? Are they apodiformes?
No, I think swifts and hummingbirds are, yeah, thank you, Sushma.
Okay, I'll chime in. Sorry.
Go for it.
So hummingbirds are grouped within the group of nightjars, carpomulgiforms, and swifts as well. And I don't remember the date either because we don't agree on some of these things, but I would imagine it's like closer to the KPG, so pretty early divergence. Yeah. So yeah, I think the common ancestor probably didn't look more like a nightjar than a hummingbird. The hummingbirds are fairly derived. So, but I really like the question that you're asking with it. I think that's a really neat way of thinking about it. Also that they have such different ecologies. I think it'd be a really neat way to test what you're doing. I had another question, if I can just chime in with that now.
Mm-hmm.
I loved your talk. I'm so impressed by how innovative you are and how you do your things. And I would love to hear more about how you have designed some of these ways of testing for things in the field. I love that you're I think you're building some of these, is it? These like ways of measuring the respirometer and other things. Could you speak a little bit more about like how much time you think about like how to design it and what you actually do to make these machines and stuff like that?
I don't make the respirometry system. We buy it. It's like $20,000 from Sable Systems. But I did not know how to use it when I first went to the field in Ecuador. And Don was just like, oh yeah, we ordered that, like take it and bring it to the field and we'll use it. And then I got there and I was like, oh, you need tubing and you need like connectors and you need all this stuff there's no Home Depot in the middle of the Mindo region in Ecuador to like go and get pieces from. So someone from Georgia like had to bring it to us. It took weeks, and then you have to import gas, and it's a lot of, there's definitely a lot of things that you still have to do after you order the system. What we built was the little chambers, because the thermal camera has some minimal focal distance and the respirometry system can't be in a giant chamber. The kind of washout time is too long. You have to, it depends on how you're running it, but we use the system where we push air in and we pull the air out. And so we can make a pretty small chamber to do the respirometry, and it has to be sealed. So we designed the chamber, and we have to adapt our design. Like with the hovering, we could do it without even catching a bird at a feeder outside. But if we're measuring thermoregulation, for example, we have to have a temperature control chamber and pull the air out of that. So we have to have a kind of refrigerator cabinet almost in the field with us. Yeah, and then the doubly labeled water, we have to come up with ways of like making a hummingbird pee in a reasonable amount of time. So we feed it a whole bunch and it keeps drinking nectar and peeing a lot. But yeah, there's a lot of variation across the different field sites. Like in Ecuador, I had to completely change what I was studying because we couldn't recapture a bird again for doubly labeled water. And then at the other cloud elevation field site, like mid elevation field site, we couldn't follow birds to look at their behavior and come up with any sort of activity budget. So we had to model it. Yeah, I don't build a respirometry system, and the
No,
thermal
but,
camera is like $100,000 from FLIR.
Right, but the ways to use it, you do have to build, and you have to spend some time thinking about and adapting. That I also think it's interesting that you have to really think about not just the organism you're studying, but where you're studying them and how you have to do that. So that's really cool. I'd love to see a video of hummingbirds peeing. I can't even picture that.
Oh, I think I have some. Yeah, I have some in thermal video. Like you can see the hot pee coming out of their bodies, and then the hot drops of pee on the chamber.
I love
So
that.
exciting.
There are a whole bunch of other questions on the chat. I should let you get to that.
Maja or Maja? I don't know how to pronounce it.
It's Maya. You got it
Maya,
right.
okay. I had a similar question about the feeders. You said that you didn't catch any hummingbirds at some locations. What are the reasons? Yeah, I did catch hummingbirds, so I was able to catch them and do experiments on them. I just couldn't do the doubly labeled water because we couldn't recapture the same bird in 24 hours. I only recaptured one bird ever actually at that site for 7 months. But the reasons were they didn't come to feeders because feeders hadn't been put up at those sites. Like there wasn't a history like there is in North America of using feeders at random locations. It was also a pretty remote site, so there weren't many people there in the first place. But the other thing about that site was it was like a high elevation site that was in between other regions, but didn't seem to have a resident species of like population of any hummingbird species. There were some territorial species that seemed to be there year round, but I don't know if they were the same individuals. Like I feel like it was a transition site between other sites, which is really hard to know until you get there. And there isn't a banding history at the site, so no one knows what individuals are which. I think that's the main reason. So we'd have to put up nets at flowering plants and find other ways to catch them instead of the feeder traps. Yeah. Colleen was asked, thank you though. Colleen was asking if hummingbirds are like lizards in allometry, do any of the genes important in hummingbird top overlap with reptiles? I would love to know that. How do you find that out? I don't know yet. Like, I'm just starting with all of the gene expression data. I have it backed up. Like that's where we're at. I made a few plots, but I don't know, and I would love to know. Yeah, if you have questions or suggestions for how to do it, please email me. What is known about, and it would be interesting to do like reptiles at higher temperatures and lower temperatures and then measure and compare that with hummingbirds. Yeah. What is known about hummingbird lifespans versus other small birds? I'm wondering if topper increases or decreases or increases senescence. That's a very good question. Hummingbirds live way longer than you'd expect for their body size. You'd expect them to live 1 to 2 years max, and they live 7 to 12. 12 is the longest known, average of 7. So animals that use topper, even bats, live longer than you'd expect, and they use topper as well. I think topper does decrease senescence just correlationally, and it makes sense because they're kind of like stepping out of time for a third of their lives if they're using topper every night. And it is thought that it might have some bad side effects. There could be some reactive oxygen species, but they could also be decreasing because their metabolism is so low. So we don't know all of the mechanisms yet, but it makes sense logically that it would help them increase their lifespan. Yeah, I love that question. And then Sam was following up on Ken's question. Do you think seasonal migration and migratory connectivity could contribute anything to seasonal differences in daily energy expenditure that you found? Were you sampling the same population across seasons and thinking about if birds that stay later have particular adaptations for that? So this was definitely the same population. This was a highly banded population, and so we could track which individuals they were. And it was not in the migratory season. It was in like June, and they migrate in early May and then later in, I think, late July, August at least, if not later. So these were the same populations of individuals. There are seasonal differences in daily energy expenditure, I'm sure, across migration and in topper use. So there's a collaborator of mine based in Canada now who is a PhD student. He's studying ruby-throated hummingbirds, which we find on the East Coast, and their topper use across seasons together with the fat that they're storing. So yeah, they seem to they seem to use torpor at a higher fat threshold when they're preparing for migration. They use it at 30% body fat when normally they use it at 4%. So they're using it as a way to increase the amount of body fat they're storing in the pre-migratory season. But daily energy expenditure wise, they're probably spending a lot during the migration. Like ruby-throated hummingbirds double their body fat when they're preparing for migration, double their body weight, sorry, they go from 3 to 6 grams. Colby has something I think I'll be very interested in. Googled a gene, you mentioned RSRP1, found some cool stuff that you've probably seen, but I'll put it here anyway. Seems widely expressed throughout human tissues as well. Can be upregulated in human tumor tissue and appears to increase resistance to apoptosis. Maybe the hummingbirds express it to limit. They don't express it though, they way under express it in torpor compared to normothermy. So if anything, it's like the opposite. They don't need to do that apoptosis resistance when they're in torpor then. But I will look at that paper, that seems useful. I clearly have not read as much as I should yet. I've like tried to set my army of undergrads on reading all these things, but they can only do so much so fast. I'm copying the link over. Yeah.
And then Anusha, I did have one question kind of following along with the torpor that I might have missed. I froze a couple times during the talk. Do you know what the like relative difference is between energy expenditure coming out of torpor compared to the other activities, you know, rest, perching, flying, hovering? Because I would just think it would be so expensive to raise your body temperature that much, and you're doing it, you know, every day, and maybe only in torpor for a few hours each day.
Yeah, I think it would be too expensive to go and come out multiple times. But relative to their total daily energy expenditure, it's negligible. Relative to their total nighttime energy expenditure, it can be a big portion, but it's still way cheaper than not using torpor. So in hibernators that are using like an 8-month torpor bout, they come out every 2 weeks, warm up and like sleep for a day. And so that seems to be the most expensive part of this 8-month bout, is that rewarming period. So yeah, that's a very good question. But I think in hummingbirds, it's not much of their daily energy expenditure. If they stayed awake if they stayed asleep at a high body temperature all night, then it would be more.
Awesome, thanks. Let's see, looks like Alex maybe has one more question there. Can you go through over and under expression of genes?
Do you mean which genes were over and under expressed significantly? Alex, was that what you meant? The genes that are significantly over and under expressed?
Yeah,
I'll try to get that up.
I think it was in relation to torpor and what you, I can't remember exactly what system you were talking about in the hummingbirds, but you were looking at the genes for being over and under expressed as a result of torpor, I think.
Oh, yeah. I can show you like a couple of plots. One sec. I don't know how late you can all stay, but let's see. I'm trying to find it. Like I have some volcano plots and stuff. Well, I can show you this one here. So this is the pathway analysis kind of stuff for genes that were upregulated in torpor compared to normothermy. Can you see the right screen? Can you see the screen with the plots on it? Okay. Yeah, so the clock genes were the most overexpressed in torpor compared to normothermy. They were upregulated. There were also these cation coupled transporter genes, interleukin genes. Yeah, a bunch of them. And then this is the same analysis with a different pathway set. So it's comparing to a different set of genes, and circadian rhythm was still the most important. But also aldosterone regulated sodium reabsorption and serotogenic synapse stuff, And then these were the ones that were downregulated. And like I showed you earlier, this keratin metabolism pathway seems to be the most downregulated in Topper, as well as these essential amino acid biosynthesis kind of things. The non-essential, I guess, if they're by being biosynthesized, phenylalanine, tyrosine, and tryptophan biosynthesis was downregulated in deep Topper. I do have the volcano plots of the genes somewhere, but it's going to take me a minute to find them.
Thank you.
Yeah.
Awesome. Well, if we don't have any other questions, then I think we can end it there. It looks like we're a little past 1, but thanks again, Anusha. That was a really, really fantastic talk. Thank you. This was so interesting. And you asked such interesting questions. I don't usually get questions I can't answer anymore, but you brought some up, which is great. I will look into them. Thank you all. Thanks again, everyone, for joining. We'll see you next week.